Welcome to the Mindfulness Series, where we bring you a variety of episodes to keep your mind full of the lessons we learn about ourselves in this podcast. Today's uncut episode features Professor Tschida, a current professor at Cornell University who brings neuroscience and psychology to parallel her discoveries in various animals with humans. This is your host, Day One, and you're listening to the Mindfulness Series. All right, thank you so much for coming on today.
Yeah, sure thing. It's a pleasure to be here.
Yeah. Do you think you could quickly introduce yourself?
Yeah, my name is Dr. Tschida. I work on mouse love songs. I say that sort of facetiously. To give a little bit more detail, I work on the neural circuits that underlie the production of vocalizations. So I started my own lab at Cornell University in the psychology department. As of this January, it's been kind of a weird time to start a lab. Just prior to a pandemic. Let's see, originally I'm from Iowa. I was at Duke University for a really long time after my undergrad. And yeah, I just moved to Ithaca, New York last December.
Yeah. So what would you say is your biggest area of study? Where have you studied? Where do you work? And any major accomplishments you really want to mention before we start our questions?
Yeah, sure. I actually got my start in science working with songbirds. It was kind of random. I was an undergraduate taking a neuroscience class, and we read this paper about the neural systems, the neural circuits for vocal learning in songbirds, and it just sucked me in. I thought it was so cool that birds have these dedicated brain regions for learning and producing their songs, and they also have these neat behavioral parallels to how people learn speech as infants and children. So my background is really in vocal learning and neural circuits for vocalization. So I did my PhD work also in songbird vocal learning. In terms of what we accomplished there, I, in collaboration with a postdoctoral fellow in the lab at the time, we developed 2-photon imaging of single neurons in the songbird, and that was really the first time that had been done. And we were looking at these tiny little structures on the neurons called dendritic spines, which are these morphological sites of synaptic input. And we were basically tracking how those change. So they go away, new ones grow, or they perhaps change in size. And you can use that as a correlate to understand how the inputs onto a given cell are kind of changing. And so in our case, we were looking at how that happened over the course of song learning and auditory experiences that are important for the bird to learn its song. So after that, I had kind of a brief detour. Another kind of big passion of mine, in addition to vocalization, is just motor control. So I worked for a while with fruit flies, and I studied the descending circuits for the control of leg movements in the fruit fly. And I love that work. And I also really miss working on social behavior and vocalization in particular. And, one really great thing about the fly, though, is that it has this massive toolkit in terms of just genetic tools that you can use to access neural circuits to kind of probe and measure their function. So in my more recent work, I've switched back into a vertebrate system. And so I work with mice now, studying the neural circuits for the control of vocalization in the mouse.
Yeah, that was great. I think you actually answered my next question, which was tell me how you got to your interest. But I think that was perfect. So, I mean, you've already done like a quick overview of like your lab and research that you've done. But do you think there's anything else that you really want to like talk about, like your ultrasonic courtship vocalizations? Maybe talk a little bit more about that, just in general perspective.
Yeah, sure. Yeah, so I can tell you a little bit about kind of, the work that I accomplished just prior to starting my own lab and sort of where we're headed and what sort of the big picture questions are that I'm interested in, the things that keep me up at night thinking. So prior to my postdoctoral work, a lot was known about the neural circuits for the control of vocalization. There was a lot of work, in particular in primates, done in the 70s. And in short, we had identified a number of brain regions that were known to be important for the control of vocalization. But it was sort of this black box type of organization. So there's like this region, which projects to this region, which projects to this region. And that was sort of, if you can envision like these black boxes connecting to one another, this diagrammatic sort of, understanding. That's sort of where we were. And there wasn't really any detailed understanding of which cell types within these areas were important for vocal control. So for my postdoctoral work, I really wanted to tackle these questions and start jumping into the neural circuits at a point that I thought was really going to be like, just a site within the brain that was absolutely essential for vocalization. So we jumped in at a part of the midbrain called the periaqueductal gray. This area, like really every part of the brain, is incredibly heterogeneous. It contributes to many different behaviors, many different functions. And so at the time that I started my postdoctoral fellowship, another postdoc in the lab had developed this really nifty activity-dependent labeling strategy, whereby, without going into the details, you're basically able to express viruses and transgenes in neurons that were recently active during a behavior of interest. So we basically leveraged this tool to label neurons in this part of the midbrain that were active during vocalization. And in short, we identified neurons that appear to act as command neurons for the production of vocalizations. And so, as I said, we're doing this in mice. So I can give you a little bit of background also just about the behavior in mice. So they produce ultrasonic vocalizations. A lot of people don't know this, and it's for an obvious reason. We can't hear them. These vocalizations aren't within the human hearing range, and you need specialized recording equipment to detect them. But they produce these vocalizations. They're very acoustically complex, and they tend to produce them predominantly in social situations. And so one really scenario in which it's very easy to elicit these vocalizations is to put a male mouse in with a new female social partner. And as that male is trying to court the female, he'll produce these vocalizations during periods of social interaction. So basically that was the type of vocalization that we were studying. And so we started out in male mice, and we used this activity-dependent tagging to express viruses and neurons in the midbrain that were active in the male mouse during the production of courtship vocalizations. And then what we found is that later on, if we expressed channel rhodopsin, which is a protein, it's a light-gated ion channel. So if you shine light onto these neurons, you can elicit activity. So if we took a male mouse, put him in a novel chamber all by himself, no social partner, no social stimuli. So this is a scenario in which a male mouse would typically vocalize very little or not at all. If we artificially activate these neurons, we could get that mouse to produce USVs, ultrasonic vocalizations on command. So the activity of these neurons is sufficient to elicit ultrasonic vocalizations. And then on the other hand, if we express a protein in these cells to silence them, to prevent them from releasing neurotransmitters to communicate with downstream cells. So if we silence these cells and then put that male mouse in a chamber with a female, he'll continue courting the female, following her, sniffing her, chasing her, but he does it silently. He no longer can produce these ultrasonic vocalizations. So that was, those two observations were really kind of the linchpin observations in my postdoctoral work, that we'd really identified a completely essential part of the neural circuit. And so in terms of, where we want to go from there, I think, the questions are in some ways fairly obvious. So one major line of research in my lab is going to be to understand really how specific versus how generalized are these cells. So are these cells really specific to the production of courtship vocalizations? Are they more general than that? Do they control the production of all ultrasonic vocalizations? Or to get even bigger yet, do they control the production of all types of vocalizations? So for example, mice squeak, right? That's the other vocalization that people think of typically when they think of a mouse. I suspect that these cells are really important for ultrasonic vocalizations, and that other cells underlie the production of other kind of acoustic categories of vocalization, but it remains to be tested. So that's one major question: how specific versus generalized are these cells? A second question is really what lies upstream of these cells. Vocalizations aren't produced in a vacuum. Animals vocalize in very particular social contexts, so they're obviously paying attention to what's happening around them, integrating, interpreting sensory and social cues, and in addition, throw in there that animal's own level of social motivation at a particular moment. And all of those things are being sort of integrated and considered, and that information has to have access to this circuit. And so basically the second line of research that will be happening in my lab is to really understand what are the upstream inputs to these cells and how are they actually triggering and or suppressing the vocalization circuit within the midbrain to enable the animal to produce vocalizations in a socially appropriate manner. And then the third line of questioning is really to go downstream from these cells. We know that these cells, there are many steps back from the muscles. So there are motor neurons that provide direct input to the muscles. There are premotor neurons that provide input to the motor neurons. And so these neurons in the midbrain are like premotor neurons. So there's a lot of stuff downstream. And again, it's kind of the same thing. Many structures, many parts of the brainstem have been implicated in vocal control, but it remains pretty much unknown exactly which populations of cells are really the critical ones. And so to dive into that question, we're basically going to consider these neurons in the midbrain and to try to understand whether they form a single homogeneous output channel, or alternatively, whether they form multiple output channels that differ from one another, both in terms of their functional contributions to vocalization and also in terms of their projections to downstream structures, who they're recruiting to contribute to different parts of the vocalization. I feel like I've talked maybe longer than you'd intended, but that's a big overview of kind of where we're headed in the lab. And that's like obviously, like the kind of big picture, decades-long plan for where my research is headed.
Yeah, no, that was completely great. And I think one thing that really stood out was like the stream analogy, which is like there's like a continuous process of like neurons activity that goes down to like even our neural networks all the way down to like just motor functions, right? So I thought that was a great analogy. And I think that kind of leads to my next question, which is, so why did you choose the midbrain to actually like study? Was it because it was at the top of the stream, like you said? Or was there anything else that interested you about that specific brain part?
Yeah, we decided to focus on the midbrain in no small part due to this wealth of information we had about the role of that structure from prior studies. So as I alluded to earlier, it wasn't understood exactly which cells in the midbrain were important for vocalization, but there was a wealth of information just generally speaking about the role of the midbrain. So prior to my work, it was known basically in any species, let's say vertebrate species that had been tested, this part of the midbrain is absolutely essential for the production of vocalization. So if an animal has a lesion or damage to this part of the brain, they can't produce their vocalizations. So basically against that backdrop, we figured this part of the brain is very likely to be something that's relatively motor related and really essential for the production of the behavior per se. And then things that lie upstream of it are probably more related to, helping the animal detect and interpret and integrate sensory cues and context and motivation and basically triggering the structure. And that hypothesis, as far as we have seen, has turned out to be absolutely spot on, but also was, kind of standing on the shoulders of giants kind of thing. Like there was a lot of prior work that would suggest that this part of the brain was going to be a really critical structure for vocalization.
Yeah. I think my next question would be kind of like what you've observed in the lab. So you said that with like specific altercations, you could actually limit or even like stop the vocalization of ultrasonic vocalizations basically, right? So do you know what the effects were, like mentally, psychologically, like in that process where the mice couldn't translate anything?
Yeah, that's a really good question. So, let me back up a step and just say, going into those experiments, it was totally possible that this part of the midbrain could play a more generalized role, not just in vocalization, but perhaps, in the case of courtship, sexual motivation. So we weren't sure when we did those experiments if we were going to just completely knock out vocalization and courtship behavior or have more selective effects. And as it turned out, we had really selective effects on vocalization, and the male mice still continued to court the females, other than the fact that they weren't producing these USVs as they did that. In terms of what the mouse thought about that, it's a really fascinating question, and it's really an active area of research. Basically, I think the question you're getting at is, are the mice aware that they're vocalizing? Are they paying attention to it? And are they kind of modulating, using auditory feedback and in some way, using that to shape? What I can say is that mice, they definitely don't learn these vocalizations. And so these are innate vocalizations, which, by the way, is, true of most vocalizations produced by animals throughout the animal kingdom. In terms of how carefully are they tuning into their own vocalizations, like I said, fascinating question and totally open. A lot of the studies out there have, if anything, indicated that they're not relying on kind of moment-to-moment auditory feedback. And in fact, mice that are deafened appear to produce completely normal vocalizations. So as far as we know, they're not really tuned into their vocalizations. It's hard to totally get inside the mind of the mouse, right? And know if that had any, to some extent, it's hard to detect something if you're not measuring it. And it's possible we weren't, maybe we weren't measuring things in just the right way to see some kind of an effect like that. But I like that question.
Yeah, thank you. So without ultrasonic vocalization, I think that's what it's called, right? Were they actually less successful than mice who were actually able to vocalize that?
Yeah, that's a great question, whether mice that can't produce USVs, male mice in particular, are less successful in their courtship efforts. We took a stab at that question in our paper, and the way we did that was to put the mice in an assay called a 3-chambered preference test. So in this assay, there are 2 side chambers, and there was a male mouse in either of those chambers, and they're semi-restrained. We kind of had them under a cage insert, so they can't get out of that side chamber. And then, so we have a female who can just freely roam. She can go in the side chambers and interact with either male mouse, or she can hang in the middle and interact with nobody in particular. And so for that experiment, we had one male mouse that was artificially muted, so he could not produce ultrasonic vocalizations, and then an intact male that could. And what we saw was that on average, females did prefer the male that could produce USVs, and that effect tended to depend on how much the male was vocalizing. So if you considered cases in which the intact male just happened not to vocalize a lot, there was kind of like a split preference. So the female was equally likely to prefer either of those males. But then in cases where the intact male vocalized at a high rate, the female was more likely to prefer that male. So that's something I plan to look into that question in more detail, because I think the piece of information that you really want to know is if you were to track the long-term social dynamics and interactions of that mouse, the actual, courtship success, what you want to know is not does the female spend more time on this side or that side, but who does the female actually mate with? So I have some ideas down the road to test that in a more naturalistic way by tracking, a female's choice, but, over a long period of time where she can choose, who she wants to spend time with and ultimately who she mates with. Yeah. So I think it'll probably turn out that the USVs are important. One thing I would say is that I don't really know of any, it's hard to think of a scenario where an animal produces a vocalization that serves no purpose. I can't really think of an example of that. I think what we can conclude is that certainly, worst case scenario, the USVs can't hurt the mouse's chances of courtship. Otherwise, there's no way that they would keep doing this, engaging in this behavior, right? I suspect, I think the most common sense idea is, yes, they probably do promote courtship either by conveying that male's level of motivation or perhaps in promoting receptivity in the female, but it remains to be seen.
Yeah. How does this, how do you think this research is applicable back to humans, our interactions, and other animals as well?
Yeah, so there are a couple of ways that I think this research is applicable to humans. And it's an interesting question, right? Because I'm studying vocalization in a mouse, and they don't learn their vocalizations. It's not speech. They don't have grammar. They don't have vocabulary. They don't have syntax, as far as anyone can tell. So I want to be really clear about that. And I think the 2 ways that this research can tell us something about humans, one, if we consider the parts of the brain that provide input to the vocalization circuit in the midbrain, and if you zoom out a bit, from even vocalization, there's just this general question of how do we as humans integrate the behavioral context, and especially the social context in which we're in order to select appropriate social behaviors at appropriate moments. So I think our work in understanding how the structures and neurons that lie upstream of the PAG encode social cues and motivation and trigger the vocalization in appropriate moments can absolutely shed light on that question, just in terms of generally understanding how social behaviors are produced in a socially appropriate manner. And, by the way, that particular ability is impaired in a number of neurodevelopmental and neuropsychiatric disorders. So that's one way in which I think our work can shed light on humans. The second way I think it could shed light is really kind of in the opposite direction, going into the stuff that lies downstream of the midbrain. So as I said, it's kind of a big mess of structures and neurons in the brainstem. And although there are some areas that have been identified that seem to be important for vocal control, it still remains largely unknown exactly which neurons contribute to vocalization and how. And the point I would like to make is that whether your vocalization is a mouse USV that's being controlled at the level of the midbrain, or whether it's human speech, which is being controlled at the level of cortex, both of those structures, cortex and the PAG, the midbrain, converge onto the same premotor networks within the brainstem. So I think that if we can use the mouse as a case to understand which and how different populations of premotor neurons contribute to the nitty-gritty of the production of vocalization, I think it's quite likely that those same neurons, we're going to see these kind of repeated motifs, like these same neurons are going to be used in similar ways across vertebrates, producing lots of different types of vocalizations.
So again, going back to your publications and all that, I know that from my readings, your lab did a lot of research on zebra finches, songbirds, and eventually mice at Cornell. Or I know you guys have done experiments with mice already, but do you remember any other animals that you worked with, and why did you choose these certain animals for the topics that you were studying?
Yeah, I've worked with a lot of different animals. I've kind of, at every stage of my career, I've switched so far. Just like to mix it up, I suppose. So when I was an undergraduate, I worked with lizards. We were studying the lizard neuromuscular junction. And so you would dissect out the neuromuscular junction, and then we were doing calcium imaging, labeling different types of acetylcholine receptors. There were some electrophysiological recordings and pharmacology, that sort of stuff. It was really cool work. Again, I've always really been interested in motor control. So I think even at that early stage, I knew that I liked motor control. But, I think in some ways, like where you start, I don't know, maybe some people are more deliberate than this, but it's just sort of like what's available and what you see that catches your eye. So I went to a small liberal arts college. I went to Grinnell College in Iowa. At the time, there wasn't technically a neuroscience major, but there was a handful of labs that did neuroscience. And so of the labs, that was the one to me that was the most interesting. So I started with lizards, and it was motor control, but in this really reduced and dissected prep, which it became clear to me pretty quickly. I didn't want to, I cared about, I want to study behavior. And so taking it back into an intact animal where there is behavior, and in particular social behavior, was really, where I headed after that. My next step, as I said before, was in some ways a little bit random. I read a paper in a class, and I just thought, this is awesome. I'm doing that. I was so psyched about song learning in birds after I read that paper. When I applied to graduate school, I only applied to programs that had a really strong Songbird lab in their program. I just was set. I knew for sure that's what I was going to do. And in some ways, after my PhD, in terms of studying neural circuits for singing and song learning, the lab that I did my PhD in was one of the best labs out there. Still is, I'd say, right at the top of the labs out there. And so it was difficult for me to figure out which group would I go to where I would be, you always want to, if you're going to stay in the same area from your PhD to your postdoc, I feel like you want to there's got to be a different spin on it, or maybe the lab just has some set of techniques that weren't available to you before. And I just didn't feel like that really existed. I felt like I was in the lab that had everything going on. So I decided to just kind of reinvent myself and just try something totally different. And so I thought about the things that I liked and the things I didn't like about my PhD work. And the one thing is tricky, and it was tricky at the time, and it is still tricky now working with songbirds, is just the lack of genetic tools compared to other model organisms. So I thought a lot about it, and I just thought it might be cool to try an invertebrate system with a lot of genetic tools, and the fruit fly was one of those, right? I'd taken a summer class at Cold Spring Harbor about Drosophila, and I just liked the class a lot. I thought they were just cool, neat little animals. Really enjoyed working with them, and that was it. I tend to be kind of impulsive in my decision making. I just sort of like, all right, I like that, I'm doing that, right? It was, in some ways it was as simple as that. And then in terms of my second postdoc, I kind of did the same thing. It was a similar process. I just tried to think about, what did I like most for my PhD? What did I like most for my first postdoc? And what things didn't I like? And I knew I wanted to get back into social behavior and vocalization. I didn't want to do it in a bird. I really loved having this arsenal of genetic tools at my fingertips. And it seemed to me then like the obvious choice was to study vocal control in the mouse. And I also just felt like there was this huge gap in our knowledge, and really just a low-hanging opportunity to study this in a mouse, because, at the time, no one had done it. It was just sitting there waiting for somebody to tackle. So that's basically the story of how I ended up in like 4 different animals at each stage of my career. But I do intend to stay with mice, by the way. I don't anticipate changing like yet again. I think at this point I'm pretty much set on working with mice. Yeah.
I just love that whole timeline of just stories though, because like I love how you just found your passion by just doing like impulsive but yet organized, like thought out actions, and you just find the things you love. That was like, I just thought that was just amazing to me. Wow. Yeah. So I usually ask professors to delve deeper into the most cited or published work. It's a little segment that I have with my episodes with professors. And according to Google Scholar, with almost 300 citations, it was rapid spine stabilization and synaptic enhancement at the onset of behavioral learning. So looking into that study, I personally did some developmental neuroscience of myself. And looking at it, I learned about plasticity and all these different type of topics. But I never really delved into like, how do you say it? Like the nervous system and like how that's affected, how behavioral learning is also like the effects that it has through like synaptic strengthening and all that, right? So before we delve deeper into the topic, do you want to give like a quick overview to the audience about what that study was about?
Yeah, I can give an overview of that study, and it's funny because it feels like so long ago. I hope I remember it correctly. It's like one of those things when you're doing work, it feels like it's so close to you. Like I could never forget these details. And then like 10 years go by, you've switched animal models, you know what I mean? And you're like, oh God, what did we do again? So that study, as I mentioned, I was working in collaboration with a postdoc in my PhD lab at the time. In fact, his name's Dr. Todd Roberts. He is the person who trained me as a graduate student. And we had developed, kind of the technical goal of the work was to develop 2-photon imaging and apply it to the bird. But the real question we are after is, what in the world is happening in the brain of a juvenile zebrafinch as it's hearing its tutor for the first time, and I'll unpack this in a second, and starting to learn its song. So things that you need to know about the behavior of a songbird. Its vocal learning is really similar in many cool ways to how humans learn speech as infants. So in zebrafinches, which is the species that we worked with, only the males produce learned songs. So a juvenile male basically hears the song of an adult tutor, which is typically its father, but in the case of an experiment, it doesn't have to be, but in the natural situation is typically its father. And there is a period of sensory learning where the juvenile bird listens to and begins forming an auditory memory of the tutor's song. And then that juvenile bird begins producing its own song, and it relies on hearing itself, so using auditory feedback from its own vocalization to iteratively shape and improve its own song. And then you can see it over the course of development approaching closer and closer to being a good match to the tutor song that it was listening to throughout development. And then finally, around the age of sexual maturation, the birds undergo a period called crystallization, where the song really stabilizes. And so zebrafinches, this process, one of the reasons this species is really great to work with to study vocal learning is that their songs are really stereotyped. So they produce these series of syllables, and in the adult bird, it's the same sequence of syllables every single time. So A, B, C, D, A, B, C, D. And so it's really easy. You know, present a juvenile bird with a tutor, and you can really readily assess the quality of copying because you know exactly what you're looking for in terms of what it should be producing. So what we're really going after in that study is when you have this juvenile bird that's never heard a tutor song before, and as it's hearing that song for the first time and starting to form that auditory memory, what is happening? What's the neural substrate of that? And we are looking in particular in a part of the brain called HVC. HVC is, you know, bird brains are different from human brains, so there's no, you know, perfectly similar structure in the human brain. But let's say for our purposes, it's a kind of a specialized song motor cortex, premotor cortex, something like that. And it's a part of the brain that's been implicated in both singing and song learning. And yeah, like in terms of which auditory inputs, basically how this auditory information that is the young bird listening to its tutor, how is that accessing the motor circuits, the sensory motor circuits that drive singing and song learning? So what is the entry point for that information? And so really, you know, kind of the more specific question of this study was asking whether there's kind of any structural correlates of tutor song learning, of that auditory memory formation when you look in this part of the brain that's known to be essential for singing.
Yeah, so I personally thought this study again was very interesting because like I had so many questions, like how does the like juvenile like bird after listening to it, like how does it make its own, like I know that they keep the same pattern as you said, like I think it was like A, B, C, and D, A, B, C, and D, but as I researched more independently, I also noticed that most of them have like independent patterns almost. Like they still have like their, I think, unique song, right? Please correct me if I'm wrong. Was that right?
No, so in zebrafinches, they do tend to do a fairly good copy of the tutor song, but you're right, it's not identical. And there are a lot of ideas about you know, why that might be, and I suspect the answer is that it's actually adaptive. So you know, a bird needs to distinguish itself from another bird, right? And so it makes some sense then that the bird it wants to copy its tutor song. I mean, this bird has mated, it's reproduced, this is a tried and true song. Like this song worked well, that bird was successful in terms of its courtship. But then it, you know, puts its own little spin on things. So if you look at the fine acoustic structure of the syllables, there absolutely are differences. So that juvenile bird, as it grows up and produces its own song, it too produces a highly stereotyped song, but it's a little bit different in the details from the father's song. You're absolutely right.
Okay. Oh, that's interesting. Yeah. So I think this was in 2012 or no 2010, something like that. It was from Duke University.
Yeah.
So over the years, as you've kind of like researched more in the subject and also you had more experience after the publication, do you think any of your findings you might change, or do you think you might want to look into maybe if you had the chance?
There were a ton of really interesting follow-up directions from that study. And I guess what I would say is I didn't have to wonder about the answers to those questions or look into them, because the postdoc I was working with, Dr. Todd Roberts, that's the focus of his lab. Like he is just knocking it out of the park in terms of asking these questions. He's done a ton of follow-up work and continues to follow up to really understand the neural substrate of that auditory memory that the tutor forms, and to understand, so one obvious follow-up question was, okay, so I can give a little bit of detail about the findings. So when we went into this story, we thought, okay, this juvenile bird, it hasn't heard any tutor song yet. So the inputs onto these HVC neurons important for singing, they're going to be really plastic and dynamic. And then it's going to hear this tutor song and everything's going to stabilize. It's going to just like sink into place, and then it's going to start learning and producing its own song. And it was actually the complete opposite. They were fairly stable to start with. And again, I hope I'm not getting this backwards because it's been a few years since I've done this work. So they started out fairly stable, and then hearing the tutor song just kicked off this period of synaptic turnover. So spines were changing shape, and they were being gained and lost, like the rates of turnover increased following tutoring. So it went from a stable to a dynamic state, which was completely the opposite of what we'd expected. And so we're looking at, from the postsynaptic side, we're looking at HVC neurons and the dendritic spines in those cells, but what are the presynaptic inputs that are actually driving that change? And so Todd Roberts has done a lot of follow-up work looking at the role of one auditory input to HVC. It's this part of the Songbird auditory system called NIF, which doesn't mean anything to anyone who doesn't work with songbirds. But long story short is that it appears to be a really, really important input in terms of conveying tutor song information to HVC and really kicking off that stabilization, turnover, and then subsequent stabilization. And what Todd has actually seen is that, and I don't remember when this more recent work came out. It's not as recent as like last year, but it's definitely not 10 years old. In a follow-up study, he basically used channel rhodopsin to artificially manipulate the activity of NIF neurons. So these auditory neurons that provide input to HVC, he's controlling their activity very precisely using pulses of light. And by driving particular patterns, temporal patterns of input to HVC through this input, he could actually shape the temporal dynamics of the song that the juvenile bird learned. So one idea, of course, there are lots of details still to be worked out, but one, that's to me was the most interesting follow-up question. And he's uncovered basically this finding that it seems like the input from NIF is really conveying timing information about the tutor song that shapes the subsequent, song learning, kicks it all off, basically.
Right. Oh, that's a really good follow-up question. Like, I didn't really know about NIF and all that, so kind of listening to that.
There's no reason you should. It's like this, it's like this weirdly named songbird auditory area.
Yeah. So I guess my final question for that study would be, I know that you said the spinal area changes also due to development, and also after listening to the songs as during the juvenile period, the sensitive period. So I wonder if that also applies to humans, because again, when I was studying developmental neuroscience, the only thing I heard about was the transformations in the brain, when we learn what happens, and never really learned about the neural circuits and all that, or not neural circuits, spinal stuff. So do you think that also applies to humans?
Yeah, I think as is the case, if you try to translate a study in an animal to, model to a human, the details are likely to be different. For example, the most obvious thing is the brain regions involved, they're not going to be the same necessarily, especially when you're talking about a bird versus a human. But I suspect that the overall sequence of events is probably pretty similar. You probably have, an infant who is, kind of gaining the ability to hear and appreciate the vocalizations of its caretakers. And I would assume that once, the infant starts really attending to and processing that auditory information, that just kicks off a period of neuroplasticity in the cortical areas that are important for speech learning. Yeah, I absolutely think, I mean, and again, the details are likely to be a little bit different, but I think that overall sequence of events is quite likely to be true.
Yeah. I had a few people in my audience who are actually very interested in this as well. So they also were interested in like motor functions, learning about animals. And one thing they asked was like, how do you actually analyze like neural circuits of the brain? So I know there's calcium imaging in vivo, sorry. There's also like viral genetics and stuff like that. Um, so do you think you could go over some major aspects of like analyzing like the animal, any
Like
different
the tools
methods,
that we
yeah, the
use.
methods and tools, yeah.
Yeah, sure. Yeah, so we tend to use similar sets of methods in many different types of experiments. And the general sequence of events is like, you want to understand how a given uh, set of neurons, it's often defined by the fact that they're active during behavior, that they express a given genetic marker, or that they project to a given brain area of interest. You want to understand how this defined population of neurons contributes to a given behavior. So the general sequence of events is you want to monitor the activity of those neurons during that behavior to see, if it is in fact related to that behavior, and if so, how. And then you want to manipulate the activity of those neurons and see whether you perturb or alter the behavior in any way. So in terms of monitoring the behavior, there are a lot of different tools out there, but the tool that my lab, that I've used in the past and that I plan to use in my own lab is calcium imaging, as you alluded to. And there are a couple different flavors of calcium imaging that are really amenable, or not necessarily straightforward, but more amenable to being applied in a behaving, freely moving animal. So one method that we use is fiber photometry. So it basically, you need to use step one is you use a virus to express a calcium indicator in your population of cells. And so the calcium it's not a perfect proxy for neural activity, but it's nonetheless a proxy for neural activity. So you're basically reading out the changes in calcium, intracellular calcium levels in your population of neurons as a proxy for the overall level of neural activity. In fiber photometry, you're implanting an optical lens just over that population of cells that expresses the calcium indicator. And a really important point is that you're measuring, again, at the population level. So you're not looking at what individual neurons are doing. You're just considering you're getting an average signal from that group of cells. And so that method tends to work best, I think, as you can imagine, in cells that are doing something kind of similar to one another. So if they tend to go up at the same time and go down at the same time, and they tend to behave similarly, fiber photometry is a great method. It's not a good method if you have a heterogeneous group of cells doing different things at different times, because everything's just going to be averaged out in the wash. And so to get single cell resolution, another method that I'm planning to develop in my own lab is called mini-scope imaging. It's basically a method where the animal wears a head-mounted microscope. So it's a single photon microscope. And so the resolution isn't quite as good as what you get with 2-photon microscopy, but it's pretty good nonetheless. And so there's usually a base plate that's on the animal's head at all times. And then right before the experiment, you snap and place this little miniaturized microscope. And those can be attached in a variety of ways. The ones that I've used in the past have been held by magnets, or perhaps a small screw. So you basically put this miniaturized microscope on right before, and then the animal just goes about its behavior. And same thing, you're measuring calcium signals from your neurons of interest, but in the case of mini-scope imaging, you actually get a field of view. So you can see all of the little cells in your field of view. And there are some algorithms that you can use to determine basically what type of a signal is a single neuron versus 2 neurons close together, things like that. So there's a little bit of trickery involved in figuring out whether you're looking at 1 cell or 2 cells sitting, very close to one another, for example. And that's obviously a little bit more difficult to implement than the fiber photometry because there's an entire microscope involved. But the great thing is you can achieve single cell resolution in a freely behaving animal. And so for the work that we're doing, this is absolutely essential because we're trying to look at vocalizations produced in social context. And it's really hard to get at that if the animal isn't allowed to just engage with its environment and social partners at will. In terms of manipulating neural activity, there are a couple of things in our toolkit that we typically use. The first is optogenetic manipulations of neural activity. So again, the starting point is a virus, and you express a protein in a cell. These are light-gated proteins that either increase or decrease the activity of neurons. And so you have to implant an optical fiber above the population of cells to deliver light directly to the neurons. But in that same way, you can manipulate the activity of those cells as the animal is freely behaving. And then the final method that we use in terms of in vivo manipulations is called chemogenetics. So this method is a little bit less invasive than optogenetics. Step one is still a viral injection. You need to express an exogenous receptor. It's a designer receptor that doesn't have a natural ligand within the mouse's brain. And then right before you want to have your animal engage in behavior, you basically give the animal an IP injection of a designer drug. And that designer drug will bind to that designer receptor that's expressed by your neurons. And there are different flavors of these things to either increase or decrease neural activity. But the time resolution isn't as nice as what you get with optogenetics, where you can precisely deliver light pulses. So the chemogenetic manipulations are better suited to things where you want to either increase or decrease neural activity kind of just chronically over a longer period of like a few hours, for example.
Yeah, and it's really fascinating to see so many different methods that you could just use for like different like scenarios and situations. Yeah. So before I go to like more general topics, do you think there's other experiences that you had in relation to like your specific work that you feel that I failed to mention?
No, I mean, I think we've basically covered the trajectory of the work that I've done. There's nothing that I was burning to talk about that we didn't already talk about. So
yeah. So you could relate this to your work, or you could just keep it general. But how do you think the neuroscience field is developing currently? Where does its future potential lie, in your opinion?
Yeah, I think I don't know if this exactly answers your question, but I think one thing that I've really seen develop in the last 5 years especially is a proliferation of methods that allow a researcher to amass a huge amount of data. So methods to either image or record from many neurons, in some cases in multiple parts of the brain at the same time. And in some ways, I feel like these methods are, the ability to get these large amounts of data is developing a little faster than our ability to know what to do with those large amounts of data. So you see, I don't mean to be critical of these methods because they're incredible, they're amazing, and surely getting large amounts of data is not a bad thing, right? Of course it's a good thing. But in terms of keeping up with the large amounts of data with actual hypotheses and questions and ideas about what to do with the data, there seems to be sometimes a little bit of a disconnect there. And I think to me, kind of, this isn't exactly answering your question where things are headed, but sort of like what where the field is going and what kind of big issues we're facing. There's this older tradition of recording or imaging from individual neurons, right? One cell at a time or a few cells at a time as an animal's engaged in the behavior. And now we have these newer methods where we can record or image from multiple sites within the brain, or multiple neurons, or like huge populations of neurons from multiple sites. And so understanding how to connect those different levels of information is something that I think, obviously, it's a huge challenge. There's no right answer to it, but I think that's one of the big challenges that neuroscience is facing right now. Like, how do we reconcile these different levels of information, and how do we yeah, what do we do with all this information we're getting about the brain, and in terms of actually understanding how brains work? It's a huge challenge.
Yeah, like personally, I thought that was really funny because I remember working in a lab and they had like literal thousands of rows of data, but like they couldn't organize it all. So like I had to do all of them and actually like translate what each of them meant. So that was just a nightmare. But yeah, that I thought that was really funny, and I definitely empathize with you on that point. Um, so these are my final questions because I know I don't really want to take up too much of your time. So why did you decide to transition your lab to Cornell and start your work there? And why do you think your work is interesting to like look into to start a career in?
Okay, so just to be really clear, I did not have my own lab prior to Cornell. I was a postdoc, so it wasn't transitioning anything. I was just starting from scratch. Um, I mean, one really honest answer is because it's where I was offered a job, right? I mean, I did have a couple of job offers, so it wasn't that I had no choice. But even there, I mean, it's like the job market. It's so tough. So to have an offer or a couple of offers, I felt incredibly fortunate. The reason I chose Cornell, one was the people. For me, like, I mean, I love what I do, but I've got to like the people that I'm going to work with every day. That's to me almost equally as important as the actual topic or the thing that I'm doing at work. So the people there are great. They're just like a really kind of the lifestyle better suits me than a lot of places that I've seen or been. People work really hard. They're awesome at what they do. And then they also are humans with lives, like it's not just about this, productivity at work. Like people are, I mean, people are really productive and they're amazing. But I think, it's sort of common sense that is your ability to be productive and creative and effective at work is sort of fueled by your time off, by having a break, by having other things that you care about and value. It just, and so being around people that held those same values was really important to me. And I was really fortunate to get an offer, from a place where that was the case. And then in terms of science, scientifically, why did I want to go there? There is just this long tradition of researchers across many different fields and departments at Cornell studying both vocal communication and just social communication in many different types of animals. It sort of has this rich history of people studying animal communication. And that was hugely attractive to me because, at the end of the day, I do study neural circuits, but I study neural circuits for communication. And to me, that, it's always, the goal is to always get back to the behavior. And yeah, finding a department where the behavior per se was really valued and of interest to people, and not just, a means to understand how the nervous system works, but, like the two things are obviously tightly interlinked, right? And so having that back and forth as part of the fabric of the group of people and the research that I'm are surrounded by was just too good to pass up.
Do you have any last messages for our audience, something that you want us to like depart with and kind of think about?
Yeah, I mean, it's hard to, there's been a lot of crazy stuff happening. It's, I feel like my last words or message are a little like, I don't know, heavier than they might normally be. I mean, I guess I would just say, I am completely just disgusted and just disgusted by the events of the last few weeks. I mean, of course, prior to George Floyd and all of these other atrocities was COVID. So there's this already very like crazy, stressed out, horrible backdrop to the situation. So what I want to just say is to those in the scientific community that have been affected by COVID, just this too shall pass. It's causing delays and stress, but I think we're all in it together. And I hope, that should be the message that we're conveying. It's okay not to be productive every second of the day. It's okay to take time for yourself. And the absolute most important thing you should be doing right now is taking care of yourself and staying healthy. With regard to the events and their impact on the Black community and people of color. I think this is especially, I don't know if you are on Twitter, right? It's just been people talking about this nonstop. And I think a lot of people are struggling right now. And again, the message, from all departments and institutions should be one of support, of solidarity with Black researchers, with people of color. We've just got to do more. I think, it can't just be single labs. It's got to make any change, these things have to come on a bigger scale. All I can say is I sincerely hope that, these recent events, hopefully some good can come out of them in terms of people actually mobilizing and making real and lasting change. But same thing there. People, I need to take care of themselves, and and I just hope that the 2020 has been a real crap year, right? Like I just hope, I just hope there's a brighter spot. This probably isn't even a good message to end on. It's like a bummer, right? It's a dark, sad message. But I hope, for me, there's hope too, that we can move forward from these things and do that together as a community, support one another and learn.
A special thank you to Professor Tschida for taking her time out of quarantine for this episode. If you want to check out her work, visit psychology.cornell.edu/Catherine Tschida. Thank you for your time, and I hope after this episode, your mind is mindful.